Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating case study that sheds light on a rare inherited muscle disease in cats. This research, recently published in the Journal of Veterinary Internal Medicine in 2024, explores a specific genetic change associated with X-linked muscular dystrophy in a domestic shorthair cat that surprisingly experienced a milder form of the condition and lived to an older age. X-linked muscular dystrophy in cats, often abbreviated as FXMD, is an uncommon disease. It's a hereditary condition stemming from an abnormality in the dystrophin gene, which is located on the X chromosome. This gene is crucial because it provides the instructions for making dystrophin, a vital protein for maintaining the integrity of muscle fibers. When the dystrophin gene is not functioning correctly, it leads to muscle weakness and degeneration. In human medicine, similar issues with the dystrophin gene cause conditions like Duchenne muscular dystrophy, which is typically severe and progresses rapidly with an early onset, often resulting in a life expectancy below 30 years. Becker muscular dystrophy, another related human condition, tends to have a later onset and progresses more slowly, with a wide range of severity. In cats, cases of dystrophin deficiency have been reported, often characterized by muscle enlargement and collectively known as X-linked muscular dystrophy in cats. The progression of this disease in cats can vary significantly between individual cases. For instance, some cats may show signs early in life and have a severe disease course, while others might develop the condition later with milder symptoms. While many different genetic variants affecting the dystrophin gene have been identified in humans with muscular dystrophy, our understanding of the specific genetic changes responsible for FXMD in cats has been limited, with only a few such variants reported previously. These earlier reports identified specific genetic alterations, like a nonsense variant in Maine Coon cats associated with a severe, juvenile onset form of the disease, and a Missense variant in a domestic cat with a milder, adult onset presentation. Another nonsense variant was also found in a domestic cat with a severe, juvenile onset condition. This highlights the complexity of how different genetic changes in the dystrophin gene can manifest in varying degrees of muscle disease in cats. This study aimed to further our understanding by investigating the genetic basis of a case of FXMD, presenting with a less severe clinical course. The study focused on a 9-year-old neutered male domestic shorthair cat, who was brought to a veterinary hospital in Tokyo, Japan, due to persistent muscle swelling and occasional breathing difficulties that had started around the age of 3. Initial blood tests revealed abnormally high levels of certain muscle enzymes, specifically alanine aminotransferase, aspartate transaminase, and creatine kinase. Subsequent physical and neurological examinations showed swelling in the muscles of the neck and upper limbs, a slow gait, and infrequent episodes of labored breathing. An electromyography, a test that measures electrical activity in muscles, revealed unusual patterns called pseudomyotonic discharges and complex repetitive discharges, which even produced a characteristic dive bomber sound. A muscle biopsy was also performed, and microscopic examination showed signs of muscle cell death and regeneration. To understand the underlying cause, the researchers performed whole genome sequencing on this cat. This comprehensive genetic analysis identified a novel and unique genetic variant in the dystrophin gene. Specifically, they found a hemizygous nonsense variant, meaning it was present on the single X chromosome of this male cat, leading to a change in the genetic code at position C.8333G greater than A. This change was predicted to cause a premature stop signal during protein production, resulting in a truncated, likely non-functional dystrophin protein. This specific premature termination codon was designated as P2778TER. Based on the combination of the clinical signs, the microscopic findings in the muscle tissue, and the identification of this specific genetic variant in the dystrophin gene, the case was diagnosed as X-linked muscular dystrophy in cats. Notably, despite the presence of this genetic abnormality, the cat exhibited relatively mild clinical signs and had lived to the age of 9 at the time of examination. Further investigation confirmed the presence of this genetic variant using Sanger sequencing, a more targeted method of DNA analysis. The affected cat was found to have this variant on its X chromosome, while control cats did not. To assess the potential impact of this genetic change, the researchers compared the corresponding region of the dystrophin gene across different animal species, including humans, dogs, cows, pigs, mice, and zebrafish. This comparison showed that the normal genetic letter at this position, guanine, was highly conserved across these mammalian species, suggesting its importance. Interestingly, the identified feline genetic variant was found to be analogous to a specific variant in the human dystrophin gene, known as PTRP2786-TER. In humans, this type of truncating variant is considered disease-causing and has been observed in a patient with Duchenne muscular dystrophy. Like the cat variant, the human change also involves a switch from guanine to adenine, leading to a premature stop codon. This specific human variant is not commonly found in the general population. Furthermore, computational analysis using a tool called MutPred-LOF suggested that the identified feline variant was likely to be pathogenic, supporting the idea that it was responsible for the cat's condition. The discussion of this case highlighted several important points. Firstly, X-linked muscular dystrophy in cats is characterized by muscle enlargement, slow movement, and breathing difficulties, often accompanied by elevated levels of muscle enzymes in the blood. While severe, early-onset cases in cats often have a poor prognosis, this particular cat presented with a later onset of signs and a milder clinical course, surviving to an older age. Although tongue protrusion was observed, it did not lead to eating problems, and the cat did not develop other complications like megaesophagus, which have been seen in some other cases of FXMD. The electromyography findings were consistent with previous reports of FXMD. The muscle biopsy confirmed muscle damage and regeneration, typical of the condition. Unfortunately, a detailed analysis of the dystrophin protein itself in the muscle tissue was not possible in this case. The occasional tongue swelling and breathing distress in this cat responded well to treatment with prednisolone, a type of steroid medication. While prednisolone is used in humans with Duchenne muscular dystrophy, its effectiveness in cats with FXMD has not been well established. This case suggests that it might have a beneficial effect in managing some of the symptoms, although further research is needed. The fact that this cat with a nonsense variant in the dystrophin gene exhibited a milder phenotype is interesting because usually, such variants lead to the absence of the dystrophin protein and severe disease. However, in humans with Duchenne muscular dystrophy, around 10% of those with nonsense variants can have milder Becker-like or intermediate phenotypes. This can sometimes be due to the production of alternatively spliced versions of the dystrophin messenger RNA, potentially leading to some truncated but partially functional protein. The analogous human variant to the one found in this cat has also been reported in a human patient as a truncating variant. Although the researchers in this feline case did not perform detailed analysis of the messenger RNA or protein, it is possible that a similar mechanism contributed to the milder signs observed. The study also touched upon the risks associated with volatile anesthesia in humans with Duchenne muscular dystrophy, which can lead to a severe muscle breakdown called rhabdomyolysis. Total intravenous anesthesia is considered a safer alternative in humans. A similar association between volatile anesthesia and rhabdomyolysis has been reported in cats with FXMD. In this particular case, total intravenous anesthesia was used for the diagnostic procedures without any complications. However, the authors caution that since this cat had a mild form of the disease, the safety of total intravenous anesthesia in more severe cases of FXMD still needs to be determined, and they recommend avoiding volatile anesthesia when FXMD is suspected. The identified genetic variant in this cat was unique and had not been found in a large database of cat genomes, suggesting it is a rare change. While the study provided strong evidence that this genetic variant was likely responsible for the cat's condition, the researchers noted some limitations, such as the unknown family history of the cat and the lack of detailed protein analysis. Despite these limitations, this study provides valuable insights into the genetic basis of X-linked muscular dystrophy in cats and highlights that even nonsense variants can be associated with milder clinical presentations, expanding our understanding of the genotype-phenotype relationships in this rare disease. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.